Then after discussion, I should go over. Yes.
You are part of the study. So let's go. So they said that. So the first question I have here, they said that which property of water is directly responsible for its high latent heat of vaporization? Okay. What do
Yes,
we
sir.
call latent heat of vaporization? What does it mean? You see, pay attention. If you hear the word latent heat, you look at this word latent heat. If you hear the word latent heat, keep in mind that there is a what? There's a change of state. There's a what?
Change of state.
Change of state. So if I say latent heat of fusion, if you hear the word fusion, what should you have in mind? Take note that we have from solid to liquid. Solid to what?
Liquid.
And if you hear vaporization, so what do we see? We have what?
Liquid to gas.
Liquid to gas. Thank you. So this is how you can decode the meaning easily. So if I'm not saying latent heat of vaporization, so I'm trying to tell you that it is the heat required to change liquid to gas without a change in temperature. Okay, that is whenever there's a latent heat, take note that there's what? No change. There's no change in temperature. Okay. Yeah, there's no change in temperature. So now looking at the options, you have it with you. So what's the answer? The said which property is responsible for its high latent heat of vaporization? Is it the covalent bond within its molecule? Is it the hydrogen bond, the high density, or its high transparency? What is the answer?
Hydrogen bond.
Okay, exactly. Remember that is due to the presence of the what? Hydrogen bond. Our answer is what? Hydrogen bond. Yeah. Remember this hydrogen bond. See, look at H2O. This is oxygen and 2 atoms of hydrogen. This is it. Okay, look at another molecule of water here. Oxygen and 2 atoms of hydrogen. Now you see that bond that's always drawn in broken line, connecting this hydrogen to this oxygen. So, connecting this to this. This is what we call the hydrogen bond. Hydrogen bond. Good. So that is responsible for its latent. And pay attention to something that water has high boiling point due to the presence of this what? Hydrogen bond. This hydrogen bond has a lot of effects on the boiling point of water and so on like that. So our answer is what? B. Is due to the presence of the hydrogen bond. Next one, question 2. They say, why is water considered an effective coolant for human eating? Why? Why is it called effective coolant? Why?
Because it requires a large amount of thermal energy to break
the what?
Bond.
Exactly. So why is water considered an effective coolant for human? Because it has what? Exactly. It is a coolant because let me tell you, because before you can make water boil, you will need high amount of energy. So it means that it is best to use water to cool. Do you know why I cannot use granite oil to cook my body and to cool my body? Do you know why? It is because if I use granite oil to cool my body, granite oil can easily boil. How together?
Yeah.
But water, you will need high amount of energy to make it work, to make it start boiling. So our answer is what? Option what? Option what? C, yes. Evaporation requires a large amount of thermal energy to break the hydrogen bond. Question 3. They said that which solute is transported in the human blood plasma, primarily bound to hemoglobin, rather than freely dissolved in water? What's the answer?
C, oxygen.
Oxygen?
Yes.
Why?
Because oxygen cannot be dissolved in water, and it is.
Let me see. Hold on. Water solute is transported in human body. Solute is transported in human body, in human blood plasma. Okay, which solute is transported? Oh, in the human blood plasma, primarily bound to hemoglobin. Okay, what's the answer? The answer is what? Oxygen. Now, pay attention to something. You see, pay attention. You see your blood cells. All together?
Yes.
Your blood cell. Remember, you have what we call the red blood cell, you have what we call the white blood cell, and you have what we call the what? The platelets. Are you there? The blood
Yes.
platelets. All together? Now,
Yes.
you see this red blood cell, it contains a pigment, and that pigment is called hemoglobin. What do I call it?
Hemoglobin. Hemoglobin.
Hemoglobin. Now, what's the function of this hemoglobin? Let me help you spell it very well. Hemoglobin. What's the function of this hemoglobin? What's the function? The
To
function
spread
is that
blood
what?
around your
It
body.
helps us what? It helps in transporting oxygen. In what?
Transporting oxygen.
Yes, transporting oxygen. Yeah, exactly. So oxygen can easily see when oxygen attached to this hemoglobin, it forms what we call oxyhemoglobin. What do you call it?
Oxyhemoglobin.
Oxyhemoglobin. Yeah. So you see, one thing that can easily attack the effect of this hemoglobin is CO. You see, this CO can easily stop the effect of hemoglobin. That is why if you are in a place whereby there is smoke, it is very, very hazardous. If you are in a place whereby, let me say, you have your generator now, and the smoke is entering your room. The smoke coming out contains CO. This CO competes with hemoglobin in the blood and convert it to what we call carboxyhemoglobin. Yeah, and that carboxyhemoglobin, it is very, very stable, and it's keep on eating your oxygen before there's no oxygen in the blood, and everybody just choke and die. So that's that for that. Let's go. Next, they said that which solute, okay, they have done that one. So our answer is oxygen. Number 4, how do cohesive properties of water benefit tall vascular plants? Yeah.
I chose B. They maintain a
Mm.
continuous column of water under tension in xylem vessels.
Okay, let's see. So looking at option B, they said they maintain a continuous column of water under tension in the xylem vessel. So if you observe that, what do we even call cohesive force? Cohesive force is that force that what? It is in molecule of the same kind. Yes, cohesive force in water, either it's like the force of attraction between water molecules. Are we together?
Yes, sir.
Exactly. So when there is cohesive force, what happens? There'll be like a continuous movement of water, either in the xylem vessels. If you look at xylem, you see we have some vascular tissues. One of them, we have the xylem tissue and we have the phloem tissue. The phloem tissue is responsible for transporting food and nutrients, while xylem transports water. Are we together?
Yes, sir.
So it maintains continuous movement of water, the cohesive force. So our answer is option B. Number 5, they said, which statement correctly compares the thermal properties of water and maintains CH4? What do we say? What
For
do we
number
say?
5, I also chose B. Water has a higher boiling point because it forms polar hydrogen bond.
Exactly, yes. If you look at this now, this type of bond, as you are seeing it, it is polar. Polar means that, see, this one has negative and this one has positive. As you can see, this one has positive, this one has positive. So what you are seeing here like this now is polar. It's polar. Let's go. Number 6. If you hear the word polar, you are having positive and negative signs. That is the positive poles and negative pole. Next, number 6, Abby. They say, what term describe substances that are chemically attracted to water and readily dissolve in it? Hydrophilic. What do we call it? What do we call it? I can't hear you again.
Oh, can you hear me now?
I can hear you now. What do we call substances that are readily attracted to water chemically? What do we call it?
Chemically attracted to water.
Number 6 answer. What's the answer for number 6?
Mm,
It's
hydrophilic.
hydrophilic. The word hydro is water. And the word philic is loving. So water loving is hydrophilic. Do you get that?
Yes.
Nice. Let's go. Number 7. Which mechanism allows aquatic organism to survive under ice during severe winter condition? Okay, what's the answer?
B. Ice is less dense than liquid water, floating to create an insulating thermal layer on top.
The answer is B, correct. Number 8. What enables water to act as a universal solvent for biochemical reaction? Universal
C.
solvent means that it can dissolve. What do you say?
C.
Number 8, B is wrong. The answer is C. So let me tell you, water, as I've seen water like this now, it is a universal solvent due to the fact that it is polar. It can dissolve more substance than any other solvent. Our answer is C, due to its polar nature. Please take note of the answer. Number 9, adhesion of water molecules is most clearly demonstrated in which biological phenomenon? You hear the word adhesion. Adhesion is like, you see, the cohesion is between the same molecule. Adhesion is between different molecules. Okay?
Yes.
Good. All right. Please hold on. Give me a second.
So what is everybody going to for the entire thing? You just put a bunch of YouTubers to come and ask with them every time. So, the Ghana is not, I don't know the time and everything. And the just,
Okay.
it feels like, so like, quick, like, oh,
Are you there?
yes, sir.
Good. All right. So we are in number what?
Number 10.
Number 10. No, number 9. We are not doing number 9 now.
No, we just, oh, yes, for number 9, I chose B.
Number 9, the answer is B. You see, let me tell you something. If you pour water to glass cup and you throw the water away, you observe that the water will, you see that the glass will become wet. Not all the water will go out. Some water will stick to the glass. You see, the form of attraction between water and that glass is what we call adhesion. You see, if you see the word cohesion, it means the same molecule. If you see the word adhesion, it means different molecules. So looking at this, you see that the answer is B. Check. Water clinging to cellulose cell wall inside the xylem plant. Inside the what? The plant xylem. Exactly. Look at it. Water clinging. Look at it now. You have your water now. And you have the what? I have the cellulose cell wall of the xylem of the plant. So the form of attraction between these two is what we call adhesion. Remember that they are different molecules now. Water and cellulose cell wall, so it's adhesion. Number 10. How is cholesterol transported in the human bloodstream? How is cholesterol transported in the human bloodstream? What's the answer?
C.
What's C?
Enclosed with lipoprotein complexes with hydrophobic core and
things. Don't worry. Some of them are questions that I've actually not taught you some of them, but don't worry, you will still learn them as we move on about cholesterol. So what do we use? How is it transported? The answer is what? Option C. Enclosed within lipoprotein complexes with hydrophobic. Don't worry, you will learn all this later. Our answer is C. Okay?
Yes, sir.
Yes, exactly. You see, if you look at cholesterol, you see cholesterol, it belongs to a group called steroid. Called what? Called steroid. And as I'm looking at this cholesterol is hydrophobic.
Hydrophobic.
Yes, hydrophobic. If you hear the word phobia, it means it's so water eating.
Yes.
So it is hydrophobic and also non-polar. Yes. So take note of that. Number next. That we are done with water now. So mark yourself. What do you get?
9 out of 10.
MashaAllah, tabarakallah. So the next topic. So everything now is on water. So this one now like this now is on water. The next one is on what?
Cells and cell structure.
Okay. Let's go.
Okay.
Next, they said, which structure is present both in prokaryotic and eukaryotic cell? What's the answer?
Plasma membrane.
Correct. If I don't want to say plasma membrane, what do I say? Cell membrane. Okay.
Yes.
Number next, what is the primary function of the nucleus within a eukaryotic cell?
Assembly of ribosomal subunits.
Yes. You see the nucleus is a region inside the nucleus. Yes. It is responsible for transcribing RNA into some ribosome. Don't worry. You see this guy, nucleus. Don't worry. You still learn all this thing deeply. Nucleus. It helps us to synthesize ribosome. Have it in your mind like that. It helps us to synthesize what? Ribosome. Okay. Since we've known that, the next question is number what? Number 13, right?
Yes.
Number 12, the answer is B. Number 13, an electron micrograph shows a cell with a high concentration of rough endoplasmic reticulum and Golgi apparatus. What is the primary function? What is the primary function of the cell? What's the answer?
B. Synthesis and secretion of extracellular proteins.
Why? The answer is not B or C.
B.
Yes. B. That's synthesis and secretion of extracellular protein. Abi?
Yes, sir.
Okay. Because it is the answer because take note that if I have rough endoplasmic reticulum, pay attention to this. If I have rough endoplasmic reticulum, pay attention. Now, this is the rough endoplasmic reticulum. You see this guy that you are seeing here? This guy is the ribosome. All together. And the function of ribosome is about synthesis of protein. All together.
Yes, sir.
So it means that if I have a cell that has high concentration of this, it means that what? The prime function of the cell is to what? Is to synthesize and secrete extracellular protein. Is that not so?
Yes, sir.
Exactly. Next. Next?
Number
what?
14.
Number 14, they said, which feature distinguishes plant cell from animal cell under an optical microscope? What's the answer? C.
Yes.
Yeah, it says cellulose cell wall and vacuole belong to plants. Next, number 15, Abby.
Yes.
What limits the maximum size that a cell can attain?
The ratio
What's
of
that?
surface area to volume. B.
Exactly, surface area to volume. Correct. Yes. If you see a if you see an organism with large surface area to volume ratio, if the large surface area to volume ratio is high, if you have this to be high, it means that organism is smaller. But us, as we are big, our surface area to volume ratio will be smaller. And take note, organisms that have high surface area to volume ratio, like let me say amoeba, you see, this organism we have, they'll be able to what? Like the diffusion will be faster in them. That's what I'm saying. The diffusion will be what? Very, very fast in them because their surface area to volume ratio is very high. And so on like that. Let's go. Which of the following is an example of an organelle with a double membrane? What's the answer?
C.
C, correct. That's what chloroplast. Have you now?
Yes.
Next, number 17. Which statement provide evidence supporting the endosymbiotic theory?
I
Yeah,
chose
what's the
C
answer?
for that one.
You did not do it.
I chose C for it.
Yes, correct. Don't worry, you still learn them. C, yeah. Number 18. Read number 18.
Number 18. Yes. What is the resolution of a standard light microscope compared to a transmission electron microscope?
The answer is A. Learn that thing. Very important.
Yes,
Learn
we
it. Very
learned it.
important. Yes. So number 19. Number 19.
19 was if a micrograph image of a cell measures 40 millimeter across and the actual size of the cell is 20, what is the magnification?
Okay.
And I chose C for that one.
Magnification of microscope. What's the formula for magnification of microscope? Is what?
It is
How do we say? We say the actual size
of
out
the
to have,
yes, divided
or
by the
no, take it other way around. That is the magnified one. That is what the microscope magnified divided
yes,
by the main
by the
actual
actual
size.
size.
That's 20. This is the actual size. Actual size. Now, firstly, convert these things to meter. What you're having here is 40 times 10 to the power of minus 3. Milli is 10 to the minus 3. Then 20 times. You see this word micro. It is 10 raised to power minus 6. Meter, meter. So meter cancels meter. Here, we cancel this and I'm left with 10 raised to power minus 3. So 0 cancels this one here too. So what you have here is 2 divided by 10 raised to power minus 3. And when this comes up as indices, you have 10 times 10 raised to power 3, and that's 2,000. That's the magnification. Magnification is 2,000, okay? That is, you will see them put something like times 2,000 like this. Aha, correct. That is, it will magnify it 2,000 times. Even, Shebi, you have phone that you use, right?
Yeah.
You have phone that you use, right?
Yes,
Hello, you have phone, right?
yes, sir.
Now go to your camera. Let's test something. Let me show you what come and picture. Go to your camera. Are you there?
Yes, sir.
Now show and just zoom your phone camera to the last zoom. What did you see on the screen? Why zooming it? What did you say? Hello.
Sir,
What did you see while zooming your phone camera?
it is going in like
Yes, what do you see on the screen? To show something on the screen. It's not showing you anything.
no. It's
Uh-uh.
just like increasing like 2, 4, 6, 8.
Yes, now my own increased to 10x. You see something like 10x like this.
Yes.
Exactly. That's the phone's magnification. Yeah. Now imagine a microscope now magnifying for you 2,000 times. Wow. It means that you'll be able to see what ordinary eye can never see, or ordinary light microscope can never see. Yes. Okay. Okay. Let's move on. Next question. Where are we?
Number 20.
Number 20. They said, what is the role of cholesterol in the cell membrane? What's the answer? The answer is option B, to maintain fluidity and stability across various temperatures. All right. That's that for that. So next one that we are going to is under what? Nucleic acid. Okay, let's go. They said, which of the following make up the DNA? Okay.
I choose B for that one. Deoxyribonucleic sugar, phosphate group, and nitrogenous base.
Good. What do you say? Yes, I can hear
Deoxyribonucleic
you.
sugar, phosphate group, and nitrogenous base.
Can you draw it?
Like DNA.
Okay, draw it and let me see the results. So far we've treated water. That's the correction to the test.
Yes.
Sawa Ikoma.
Alaikum salam to Professor.
Yes,
Well
ma'am.
done.
I'm
Aisha, well
currently
done.
recording this,
Salam
so I
alaikum.
will send it across.
What did you say?
I'm currently recording this, so I will send it across. So, and the corrections also. I'll send everything to the group.
Okay. All right. No
Yes.
problem.
Yeah.
Well done, Aisha.
Yes, ma'am.
All right. How you doing? How are your siblings and your mom and the dad?
Yes, fine.
Barakallah. Yeah. You're also on holiday, right?
Yes, sir.
Okay. All right. Take care.
Mhm. Okay, so draw the nucleic acid. Let's see. And remember that DNA is a polymer of nucleic acid. Yes. Many repeating units of that nucleic acid will form what we call the DNA. So what do we have?
Phosphate group.
Ahem. This is all they're saying. Yes, you say phosphate group. After that, what do you say again?
Oh, sugar. Phosphate group, nitrogenous base, and deoxyribonucleic sugar.
So this, your phosphate group. All together? This is
Yes.
your what? This is your what? Your
Phosphate
pentose. Out here. Exactly.
Are you
This
sharing
is
your screen? I cannot see it.
okay, can't see my screen again. Wow.
No.
Oh, sorry. All right, so this is it. So this is the base. This is the base. And this is the what? What do you call this? This is the sugar. If you observe the having 5 sites. So this is an
Yes.
example of the pentose
Yes.
sugar.
Sugar. Authentic sugar.
This is the pentose sugar, exactly. And that's the what? The phosphate group. Okay, good. Let's go. And remember I told you that what? If you have RNA,
I don't want
the nitrogenous base that you are going to be having, the nitrogenous base that you are going to be having is what? Can you tell me? Can you remind me? You'll be having what? Adenine, uracil,
adenine, uracil, thymine, and guanine.
guanine. This is what you'll be having. That nitrogenous base. But assuming it is DNA that you're having, what do you say you do? What do you have? Adenine. So instead of uracil, you have thymine. Have you seen it?
Yes.
Then you have cytosine and you have what? Guanine.
And guanine.
Exactly. You can see. So it's just like what is different between them is this guy. In the RNA, you have uracil. In DNA, you have thymine. So that's just what is different between them. Okay, nice. And remember, RNA is just one strand. DNA is what? Double strand. And the shape of DNA is double helix. Okay? Your DNA is double helix. So this one like this now, one strand. And this is what? Two strand. Exactly. Okay, and so on like that. All right, let's go. And remember that they are always connected by what? Hydrogen bonds. So let us just go. Let us continue some of the question. So under nucleic acid, so the number 21. Which components make up a single DNA? Deoxyribose sugar, phosphate group, and nitrogenous base. Exactly. Deoxyribose sugar. These are what? Deoxyribose sugar. And that sugar is a pentose sugar. I think that it is a 5-carbon sugar. That is, it has 5 carbon. Exactly. Okay, that's option B. Number 22. What type of bond connects adjacent nucleotide to another nucleotide?
Phosphodiester carbon.
What do you say?
Phosphodiester bond.
What are you saying? Remember, I told you what connects one DNA to another DNA?
The hydrogen bond.
Hydrogen bond. In fact, let me tag a note on the group. Go and check the group. You will see the diagram there. Open that image I tag. Like, click on that dot. It will take you back to a previous image on May 29, 2026 was when I taught you. Look at that diagram. You will see that one nucleotide and another nucleotide, a type of bond is joining them together. What is that bond?
Hydrogen bond.
Hydrogen bond. Good. Exactly. Hydrogen bond. Nice. Next, we move. So number 23, they said, if a double-stranded DNA sample contains 28% cytosine, what is the percentage of adenine present? Okay, pay attention to it. See, the question is telling you, pay attention. The question says that, what's the question? What's it saying?
Two-stranded DNA sample contains 28% cytosine. What percentage of adenine is present?
They say that a double-stranded DNA sample has 28% cytosine. So the percentage of cytosine is equal to 28%. So they said that what is the percentage of adenine? You have to find the percentage of adenine in the DNA molecule. So how do we do it? Now pay attention. Pay attention. Now, there's a particular rule you have to learn. You see that rule is called Chagas rule. Pay attention to it. It's not hard. This rule is telling you that percentage of cytosine is always equal to the percentage of guanine. Remember that in a DNA molecule, altogether, in a DNA molecule, cytosine and guanine get together. So just remember, percentage of guanine and cytosine are equal. So since they are equal, according to this question, they'll be equal to what? 28%. See, 28%, so 28%. Altogether.
Yes, sir.
Now, after that, it means that the total C plus G, so therefore, cytosine plus guanine will give me 28 plus 28, and that is 56%. Altogether.
Yes, sir.
Good. Yeah, and don't forget that adenine plus thymine plus cytosine plus guanine, everything will give me 100%. Altogether. So
Yes.
it means that adenine plus thymine will be what? 100 minus 56. These two are 56%. So therefore, what do we have? These two are 56%. So it means that 100 minus 56 will give me 44%. So therefore, the percentage of adenine now will be 44% divided by 2, and that's 22%. That's the answer. Do you get that?
Yes, sir.
Good. That's option A, Abby.
Yes.
Good. What structural characteristic explain why DNA strands are described as antiparallel? The answer is B. Don't worry, you will get here. These are still like higher parts we've not got into. All together?
Yes.
You know about this direction. Don't worry, don't rush. Let us still move. Next one. Which enzyme is responsible for unwinding and separating the double-stranded DNA? What's the answer?
C.
What's the answer?
C.
C, exactly. That is what helicase. So the enzyme is what we call what? Helicase. Nice, helicase. Good. All right, let's go. Because normally, if you have the DNA molecule like this, you see them joining together. So that enzyme is going to be like, come on, like a zip, like you're just opening something. Let me see how. Just before it gets a shot. So the enzyme will start removing this. So that enzyme is helicase. Let's go next. What do you have? Number 26. What is the key conclusion of Michelson star experiment using the 2 nitrogen isotope? What was the outcome of the experiment? Is to what?
DNA replication is
Don't worry, you get
semi-conservative.
this point. Don't worry. I have a student that have taught all this, but I did not just record the class. Don't worry. Okay, you learn about conservative, not conservative. Don't worry. So it's to show that DNA replication is semi-conservative. So DNA replication is what?
Semi-conservative.
Yeah. Okay, you get it. Number 27. The nitrogenous base are classified as what?
Adenine and guanine.
Hold on, I'm coming. Nitrogenous bases are classified. Which nitrogenous base are classified as purines? Yeah, what do you call it?
Adenine and guanine. B.
Adenine and guanine. That's question 27. Adenine and guanine. Next. What do we have? Number 28. So we have 28, 29, 30, okay?
Number 28.
What forms the structural core of a eukaryotic nucleosome?
A.
Besides what? A.
DNA molecule.
Yes, A. You will get to all this point. Number 29. In RNA, which base replaces thiamine? We have done it here. What's the answer? There are 10 years. You can see exactly. You can see. Next. Okay, what's the answer number 30?
What
B,
is the
5 to 3 prime direction. What's the direction of DNA synthesis catalyzed by DNA polymerase during replication? This about DNA replication, you will learn that. Our answer is option B. Like after this class, I think our next class is when you are going to learn this actually. Let's go. Next one is virus. Next one, we are looking at virus. Viruses. Oh, yeah, next one. If you're able to do all this, let's go. They said, why are viruses considered non-living things, non-living entities in classic biological classification? Why?
B? They lack
Because
structure and independent metabolic machinery.
they have to depend on living organisms. So the answer is B. 32. What constitutes the protein coat surrounding the viral genome? What's that? The protein coat.
Capsid.
Capsid. Are you sure?
Yes.
Yes, B, yeah, capsid. Number 33. Which enzyme is unique to retroviruses, allowing them to synthesize DNA from RNA templates? What's the answer?
C. I chose C for that one.
Reverse transcriptase. Don't worry. Number 34. What occurs during the lysogenic cycle of a bacteria? Okay, yeah, we learned this lysogenic. Okay, like I supposed to teach you one more cycle altogether.
Yes.
Okay, don't worry, leave that. But let me tell you the answer. Number 34. Let me read number 34. Take note what happens to this virus in this lysogenic cycle. What happens? The viral DNA, what does it do? It integrates into the host, what? Into the host
Host DNA.
bacterial chromosome. Yeah, exactly. As a what? As a prophage. Yes. All right. Our answer is option B, 34, B. Let's go. 35. From where do enveloped viruses typically acquire their outer lipid envelope? They are derived from what? Host cell membrane during viral budding. They are derived from what? The host what?
Host cell
Host.
membrane.
Number 36. Almost done. Which structural feature is characteristic of a bacteriophage? We have done this type of virus. I drew all of them for you, right?
Yes.
Exactly. So which structural feature is characteristic of a bacteriophage? Yeah, what do you say?
B.
What's B?
Icosahedrali
Yes.
attack.
Let me still look for the diagram and draw it for you. Did I send the note to you?
No.
I did. Hey, no. I will tag it for you now. You will see. On July 19th. Thank God I'm always sending these notes.
Okay.
Because just gonna say that he had not to. This is it. I sent it to you. Look at it. That bacteria, look at it. This is the virus. So this is the virus they're telling you about. This is the bacteriophage. Exactly. So if you look at the head, you will see what they are telling you. Now check. They said, what is the structural feature of a bacteriophage? So the answer is what? Icosahedral egg with a helical sheath and tailor what? Fibers. All together?
Yes, sir.
The icosahedral egg. So now 37. That's 36. 37. What determines the specific host range and tissue tropism of a virus? That's question 37.
Number 37, I chose B.
Antibiotics, I'm coming. Okay, no, not 37. Okay. Binding specificity between viral surface protein and host. Okay, that's correct. Number 38. Why are antibiotics ineffective against viral infection? Remember, I told you that time that was antibiotics. You will see antibiotics.
For bacteria.
Not for what? For virus. For virus, it does not work. Yeah. So antibiotics target what? Bacteria or cellular structure. You see, what this all work for is what? Is bacteria. It was for what? Bacteria what?
Bacteria.
Bacteria cellular structures. Exactly. And not for virus. Okay. So antibiotics target bacterial cellular structure and metabolic pathways. And what? Metabolic pathways. But the thing is that virus don't have this virus lack metabolic pathway. Metabolic what? Pathway. So there's no way antibiotics want to work. Next, what is a prophage? A prophage is what? A bacteriophage that's how it's B, sorry, B. A bacteriophage genome integrated into the host bacterial chromosome. B. Number 14. According to current evolutionary hypothesis, how might virus have been originated? B. As mobile genetic elements that escaped cellular organisms. Read more on virus. You have textbook with you. Okay?
Yes, sir.
So that is that about that. So let me forward this note to you. I'm coming. Wait, there's a note I want to send. Okay. Hold on. Uh-huh.